Now that we finished that, let's discuss a little bit about these definitions. So what is DNA? It's those nucleotides that are put together to make nucleic acids. So you're going to have this sugar phosphate backbone, so deoxyribose and phosphate backbone, and then you're going to have all these bases that are complementarily based pair to each other. That's all DNA is. What is chromatin? Well, remember what I said. You don't just put this willy-nilly in your nucleus. It gets wound around these spools. So the DNA gets wound around these things like this so that it's nice and organized. So it's not just thrown together like my office desk. That is called chromatin. It's DNA and these things called nucleosomes, which are proteins that are like spools. Then what happens is these will condense. This will just keep condensing, keep condensing, keep condensing until it turns into something called a chromosome. That is your DNA right here. So here is all the DNA. Now we have 23 chromosomes, got 23 chromosomes, and we could draw them all out. There's 1, there's 2, there's 3, there's 4, there's 5, 6, all the way all the way down to like 23, which is our sex chromosomes. So you see here though, something that you might notice is like, well, no, that's not 1. I'm like, yeah, well, it is. That's 2, 3. You're like, what do you mean? Well, you have 2 copies of each gene, or I mean, sorry, of each chromosome. So this is chromosome number 1 that you got from mommy and that you got from daddy. You got from mommy and you got from daddy. So you get 2 of each. So we have a total of 46 chromosomes, but that is in 23 times 2 because you get 1 from mom and 1 from dad. Well, why would we do that? Well, it's so you have a backup, right? So say I've been teaching this for so many years and I can never remember where hemoglobin is or which chromosome it's on. And I should probably look it up at some point, so I'm giving you guys the right info. But let's imagine it's on number 1. So remember, the whole DNA is not what codes for it. There's a small section of DNA that is going to be the instructions for making that protein. So let's just say right here, this small set of bases. This is where the recipe is from mom. That's mom's recipe. And here's dad's recipe. And you know mom and dad can cook the same meal. They can both make like spaghetti and meat sauce. And mom's is way different than dad's. Usually one's better than the other, right? My kids know that dad is the best cook in the family here. And that's the same thing for your protein. So mom's hemoglobin might look like this, and then dad's hemoglobin might be just slightly different. Maybe dad has sickle cell. And so he's got sickle cell, but if mom doesn't have sickle cell, then your cells can be normal. They don't have to be sickle cell. This is called the diploid number of chromosomes. There's a fancy word for the double. Diploid, double. This is 23, is called the haploid number of chromosomes. So, what's DNA? DNA is this, all bajillion base pairs, right? 3.8 billion. When it starts spooling around proteins to be organized, that is called chromatin when it starts condensing. Then eventually what happens is the chromatin condenses so much and it goes into areas of the cell where we can find it. Those are called chromosomes. And a chromosome, we have 23 of them. We have 2 copies of each chromosome, 1 from mom and 1 from dad. And then on the chromosomes, it's going to be small sections of DNA that are going to code for all of our proteins. Those are called genes. Okay, now you're talking to me right here. You're like, what the heck is this? All right, just some definitions to straighten out before we get into mitosis. What are these things called right here? These are called chromosomes, right? They're called chromosomes. These are called homologous. Homologous. What does homologous mean? Well, what does homo mean? Same. means same. So they are the same chromosome, 1 and 1. Now they're a little different because mom and dad's genes are a little different, but they have the same genes on them. Whatever the protein is to make hemoglobin is on there, both on mom and dad. They don't have different genes. They have the same genes that they have the same recipe books on all of them. Like one has this, one from mom, one from dad has the same recipes. They're just like, maybe one puts lemon, maybe one puts lime. Same thing with chromosomes number 2. You got like beef stroganoff. Maybe one uses egg noodles. Maybe one uses some sort of other flat noodle. They're the same recipe, just a little bit different. 3, you got ceviche. Maybe one puts jalapeno, one doesn't. Same thing, ceviche. So those are homologous chromosomes. So you need to remember that word. Now, what is a chromatid? Well, that brings us to mitosis. So what is mitosis? Mitosis is when we make a copy of a cell. So if you think about this for a second, your skin here is constantly dividing and it's constantly making new cells, because if you go like this, dead skin's flying off. And so you're constantly replacing that. You're constantly replacing and replacing and replacing. So if you, you learn about the epithelial tissue, right, your stratified squamous epithelial tissue, you've got this basal layer that's mitotically active, and it's going to keep pushing all of these cells off. Well, where do all of these cells come from? They come from a previous cell. So you'll have one cell with 46 chromosomes that is going to make 2 new cells with 46 chromosomes in. Does that make sense? So this is where a lot of our cells come from, that divide, that undergo hyperplasia. But what happens here is, imagine if we divided this cell in half, and then we divided the 46 chromosomes. How many would go into this daughter cell? If we did it in half, there would only be 23. And there would only be 23. Do we want this? No, we don't want this. We want to have 46. So what do we have to do to make this happen? What do we have to do to get 46 in here and 46 in here? The answer is staring you in the face. Replication. So how do we do that? Well, we have to go through this cell cycle. We got to go through this cell cycle. So we're going to have a cell. It's going to have its 46 chromosomes in it. So like, just imagine there's chromosome number 1, chromosome number 2, chromosome number 3, chromosome number 4. Let's imagine that this is a 4 chromosome. You know what? Let's make this even simpler. Let's imagine that this is a 2 chromosome organism. Here we go. 1 and 2. It's only got 2 chromosomes. All the genes to make all the proteins to survive are on these 2 chromosomes. You have chromosome number 1, you got chromosome number 2, you got 1 from mommy, you got 1 from daddy, you got 2 from mommy, and 2 from daddy. All right. So if we were trying to divide this cell equally and everybody gets equal amounts of DNA, this is what it would have to look like. We'd have chromosome number 1 here, 1 from mommy, 1 from daddy, and then we have chromosome number 2 right here. It's in the nucleus. Same thing over here. You get chromosome number 1, chromosome number 2. But if you look at this, if we divided this in half, what would we get? We'd get only 1 from mom, and we get only 2 from mom. So we'd only get, for chromosome number 2, we'd only get 1. So that would only leave us with half of what we need. So how do we end up in mitosis with the same amount that we started with? Well, what you have to do is you got to replicate all of the DNA that's in there. So that is what is called the synthesis phase. So if you think of this as a cell cycle, you think of the cell cycle like this. There is this phase of the pie right here. This is called the mitotic phase. That's when it's dividing. The rest of this is called interphase. And there's 3 phases to interphase. Can I do this right? Can I do this right? Yeah, I did this right. There's G, there's S, and then there's G2. So G is just preparing, doing normal activities, making its proteins, all that stuff. S, we've decided, hey, we're going to divide into 2 other cells. So what are we going to do with this 2 chromosome number organism? Well, let's say we have, what's the diploid number of this organism? The diploid number of this organism is 4. That is equals, oh man, somebody's screaming. What's the haploid number? The haploid number is 2. So we want to end, what do we want to do? We want to have 1 division, and we want to end with 2 daughter cells, 2 cells, with a diploid number. So I'm sure some of you have been saying this for like 10 minutes now. You got to replicate all of the chromosomes. So we have to make 2 copies of mom's number 1, and we have to have 2 copies of dad's number 1. Then you have to make 2 copies of mom's number 2 and dad's number 2. Exact copies. So if you looked at these, the DNA base pairs, 3.8 billion base pairs, they're all exactly, or however many base pairs are in that chromosome, they're all exactly the same on those. Same thing with dads. Those are exactly the same. So that makes them, the word for these is sister chromatid. What is a sister chromatid? It's an exact copy of a chromosome that is attached to, that is made during S phase. And why do we do it? So that we can have enough. Now look, you got 1, 2, 3, 4, 5, 6, 7, 8. If you divide that, you take 1 from mommy here, mom's number 1, and you take another one from mom's number 1, and you go over here and you put that there. You take dad's number 1, you put that right there, and you take dad's number 2, or dad's number 1, and you put it there. Now you have an equal number of chromosome number 1 on the left side and on the right side. Now you take chromosome number 2 from mom, chromosome number 2 from mom goes over to the other one. Chromosome number 2 from dad, you go over here, chromosome number 2 from dad. Now you have divided the cell equally. I know that's confusing, but that's the difference between a chromosome and a sister chromatid. Sister chromatid is an exact copy. A homologous chromosome, they're both the same chromosome, they have the same genes on it, but they might be slightly different. So now let's look at the steps that we have in mitosis. So in the cell cycle, first phase, we're just doing normal metabolic activities. We just replicated all our DNA. So we had 46, and if you divide 46 into 2, what do you get? 23. We don't want that. What are we trying to look for? We need 46. So if you double and you make a copy of every single chromosome and you add those sister chromatids to every single chromosome, you'll get 92 chromosomes, 92 sister chromatids. Now, if you divide that in half, what do you get? You get 46. And then the G2 phase is preparing to divide, making sure everything is replicated perfectly. Now it's ready to go into mitosis. Now, there are 4 phases of mitosis. You can remember this as PMAT. PMAT. You have prophase, metaphase, anaphase, telophase. Now, this is like being a kindergarten teacher. Anybody ever be a kindergarten teacher here? When the students, I watched a video of a kindergarten teacher one time. Every student that came in his door one day, he stood in front of them and he said something special. That's what you have to do about these, is that you have to look at prophase and say, okay, what's special about prophase? So prophase, and we're gonna do another 2 chromosome organism to do this. So I'd be able to draw this out, or I'd be able to say it. So what's the purpose? Let's go back a second. What's the purpose of mitosis? Mitosis. What cell lines are we talking about? We're talking about somatic cells that undergo this. What is that? Everything but your sex cells. Skin cells, intestinal cells, anything that divides. Not all cells divide, like your neurons don't divide, your muscles don't divide. They're in what's called the G0 phase. So this is somatic cells. All right, that's step one. What are we starting with? A diploid number. How many divisions are we doing? One division, one time. And what are we ending with? 2 diploid daughter cells. So we're going to start with this. And we're going to end with 2 of these. They're exact copies. So now what do we need to do? First off, we got to go through S phase, we synthesize, and we make copies of all of the DNA. Now we have enough genetic material to divide into here. Now we got to go through the process of dividing it. Okay, first phase, we have the nuclear membrane disappears and the chromatins condense. So when you see prophase come up, like, prophase, I'm so proud of you. Your chromatin has condensed and your membrane has disappeared. Great, you're ready for the next phase. So now the next phase is metaphase. No one should get this one wrong on a test. Metaphase means middle. M is middle. So we have the sister chromatids. That's chromosome number 1 from mom. This is chromosome number 1 from dad. Chromosome number 1 or chromosome number 2 from mom. Chromosome number 2 from dad. These are the homologous chromosomes. The ones that are attached together are the sister chromatids. Okay, lines up in the middle. Metaphase. And you can see the spindle formations. You have the centromeres, and you have these spindles that they're going to ride on. Next phase, I call this anaphase. I have 2 little girls, so I've watched Frozen more times than I care. Anaphase. Ana is when they're singing the song, "Do you want to build a snowman?" They're separated. So anaphase is what separates these sister chromatids. So this sister chromatid on the left is going this way. This sister chromatid is going this way. Anaphase. And they separate to either side. That's anaphase. It's metaphase. Now what do we have happen? Now we have telophase, and you have the T, so you're going to see the cleavage there. Now we've got 2, 4, 1, 2, 3, 4. And the last step is called cytokinesis, and that's when you separate this into 2 diploid daughter cells. That's it. That's all mitosis is. Don't get stressed out about it. It's 4 steps, and there's just a couple of words you need to know for each step. You go PMAT, I got it. I know it. I know the order. Prophase, metaphase, anaphase, telophase. Another student showed me last year, and I can't remember. Got prophase, then you have metaphase, you have anaphase, and then telophase. Oh, man, I'm gonna have to ask her to show me again. Prophase, metaphase, anaphase, telophase. What happens in prophase? You draw it. Nuclear membrane disappears. Chromatin condenses. What happens in metaphase? Middle. M, middle. Chromosomes line up in the middle. Anaphase, Ana and her sister separate. The sister chromatids separate. Telophase, you see cleavage. You see cleavage in here. This starts to cleave. Then what happens? Cytokinesis. What do you start with? A diploid somatic cell. How many divisions do you have? 1. What do you end up with? 2 diploid daughter cells. That's the cell cycle. So what do you got? You got 2 parts in the cell cycle. You got the mitotic phase, you got interphase. G, S, G2. Normal cell metabolism, replicating all of the DNA, making sister chromatids, preparing to divide, dividing. What happens in mitosis? PMAP, prophase, metaphase, anaphase, telophase, cytokinesis. What do you start with? One somatic diploid cell. What does somatic mean? Just all your regular cells that aren't your sex cells, anything that divides, not your sperm and your egg. How many divisions? One. What do you end up with? Two diploid daughter cells. Make sure you know the phases. Oh, sitting here drawing these ugly pictures, and I got them in the notes here. Here you go. So you can see in the notes. This is called interphase. Is this all of this except for the mitosis phase? When you see a cell in interphase, it just looks like a normal cell. When it goes into prophase, or when it goes into the phases of mitosis, so let's see right here. I'd say this is probably I don't see prophase in here. Oh, this is prophase, like right here. Chromatin condenses, nuclear membrane disappears. Then what do we got? We got metaphase. Don't see a metaphase in here. Why is there no metaphase? Metaphase when they line up in the middle. What is this phase right here? What's this guy? That is anaphase, right? Anna and her sister separate. I drew all these terrible pictures and they have them in here. Okay, so make sure that you know that.